Hey everyone, here's the nervous system review. What we'll be doing here is going over the stuff that is kind of required for us to know in Mark Nielsen's anatomy class. So let's jump right into it. If you do have any questions, feel free to leave a comment or send me an email. You can reach me on Canvas as well. Alright, so jumping into it, we're going to look at the divisions of the nervous system. So, as we know, the nervous system is going to be divided into different structural components. The first one we're going to talk about is that central nervous system, which is composed of a brain, and we have that spinal cord. And this picture is from the cadaver specimen in our lab. So, those are the structures of our central nervous system, brain, and the spinal cord. Then we move to the peripheral nervous system. A lot of times you'll hear people call it the PNS. It kind of sounds like they're saying penis. But the PNS has structures to it. We have these nerves, which we're going to call cranial nerves. These are all found up at the cranial end, so up by our brain. So if you can remember, one structure of our central nervous system was the brain. The nerves that come off the brain are called cranial nerves. And then the other structure of the central nervous system was that spinal cord. So we have nerves that come off of the spinal cord called spinal nerves. And these are those nerves that we learn in this class, pretty in depth. So let's look at kind of what's going on here. We have the peripheral nervous system, which is going to send a signal in to that spinal cord. So that red is representing a sensory neuron, information going in from the peripheral nervous system to the central nervous system. Once it gets to that central nervous system, we're going to have some integration. That's where we can make decisions, where it becomes conscious as it goes up to the brain. But between that sensory neuron and the next neuron in the spinal cord, so this next neuron, we have this blue. Between those two neurons, we have something called a synapse. Basically, in this is showing a basic reflex. So we have a sensory neuron going in and a motor neuron going out, and they're communicating with each other via a synapse. That takes place in the central nervous system. So that's just the basic idea. Peripheral nervous system is our input and output system. The central nervous system is our integration center. So now we're going to look at divisions of the PNS, or peripheral nervous system, our input and output system. We have something called the somatic nervous system. In the somatic nervous system, if you can connect somatic nervous system to skeletal muscle control, that's the big thing we need to know. The somatic nervous system is going to control all of the skeletal muscle in our body. And we know that skeletal muscle is controllable, and we can see this guy here. He is moving all his individual fasciculi. Pretty attractive. But yeah, somatic nervous system controls skeletal muscle, and it's voluntary. We have conscious control over this. The next part of that peripheral nervous system is something called the autonomic nervous system. So the somatic nervous system controlled everything, all the skeletal muscle, so it was voluntary. The autonomic controls our automatic stuff. And anything that's automatic in our body, specifically the muscle, is not, the automatic muscle is that smooth muscle, and it's not going to be under our conscious control. So the autonomic nervous system has two subcomponents to it, two subcategories, and we call them the sympathetic and the parasympathetic. These kind of act as opposing nervous systems, but they're going to also act in harmony to keep things in homeostasis. So we'll see some common traits here, and then some differences. So these next numbers here are important for us to know. These levels are representing spinal cord levels. So our sympathetic nervous system is going to exit, or the motor neurons are going to come off of the spinal cord at the spinal cord levels T1 through L2. So that's representing 14 different spinal levels. That's T1 through T12 plus L1 and L2. So any autonomic neuron that comes out of those levels that's going to smooth muscle is going to be a sympathetic neuron. And we'll get into more detail with that. And then on the other hand, we have parasympathetics, which are going to arise from the vagus nerve. So the vagus nerve is one of the cranial nerves that comes off of the brainstem. So that's one of our really high cranial nerves. And then when we are at the levels S2, S3, S4, those motor neurons that come out of there that are going to smooth muscles, those are going to be parasympathetic. So the key thing we need to know is sympathetic output comes from T1 through L2 levels only. Parasympathetic output comes from the vagus nerve or S2, S3, S4. And those are the key levels we need to know. So what are they innervating? Well, both sympathetic and parasympathetic are going to be innervating smooth muscle. So anywhere we have smooth muscle, it's going to be taken care of sympathetically or parasympathetically. So where do we have smooth muscle? The majority is in our blood vessels. So we've got a lot of blood vessels. The sympathetic is going to innervate that smooth muscle of a blood vessel, which causes constriction or dilation of the blood vessels, depending what we need to have happen. The parasympathetic innervates the smooth muscle of the gut tube. So that's going to induce peristalsis. Same thing with the urinary system. It innervates, basically think it innervates any tubes of our body that are not blood vessels. Alright, so now we are going to look at different types of neurons that we're going to see in that peripheral nervous system. So the first one we see is going to look like this, one of Mark's drawings. We're going to call this a multipolar neuron, and we'll go in more depth of where we find these and what they do. We also have these types of neurons called pseudo-unipolar, and then finally we have bipolar neurons. So multipolar neurons, which we're going to look at here, they're going to look like this. So that area in yellow is going to be the cell body, and everything coming off of it has different names. Multipolar neurons are going to be found in our motor neurons and neurons called interneurons, and we'll talk about those. So motor and interneurons have this shape to them, or this structure, a multipolar neuron shape. So like I said, this yellow part is the cell body. That's where the nucleus is. Here we have these little projections that are leading to the cell body called dendrites. So these can receive signals and take it to that cell body, and then it moves down this next long part called the axon. So the way the signal goes here is from the dendrite to the cell body, then to the axon. So here's a little more detail. They're going to be our skeletal motor neurons, and we have these other ones called pre and post ganglionic motor. So skeletal motor is going to go to skeletal muscle, which innervates, or which is in the somatic nervous system. And then our autonomic nervous system uses pre and post ganglionic motor neurons. And when we draw them in this class, this is how we draw them. So there's our round circle representing the cell body, and we have the long axon, and then at the end we always want to end with a fork, because that's where a synapse can occur between this neuron and whatever structure it's trying to affect. Okay, so the next one we have here, we call them pseudo-unipolar. Pseudo-unipolar are going to look like this. They are sensory neurons. So this is like pain, temperature, light touch, vibrations, discrimination, stuff like that. So any type of sensory like that. Kind of the same structure here, just modify it. There's the cell body, hangs up like a little a little sucker, little balloon there. We have this end, which we call the receptor terminals. So that could be like at the tip of my finger, tip of my toe. This is where we're going to have those transducers, um, basically just receptors letting us know there is some type of sensory input coming in. These are similar to the dendrites. This part is called the peripheral process. So we name it that because it's out in the periphery, meaning it's going from that cell body out to my fingertip or the tip of my toe or the front of my chest, just somewhere in the periphery. We have this next part here called the central process. So from the cell body that way in the blue, that's leading us to the central nervous system. That is taking the input in to the central nervous system. So it's named the central process. So we have the peripheral, which is going somewhere peripherally. Then we have the central process going to the central nervous system. And at the end, there's our axon terminals. Again, that's where a synapse can occur. That sensory neuron will talk to motor neurons and interneurons. And in this class, this is how we're going to draw those. So there on the left, we have a fork. That's where our receptor terminals are. That's going to sense we're touching something, basically. And then that signal moves across to the right to the axon terminal, where that will be in the central nervous system and communicate with a multipolar neuron. And then finally, we got our bipolar neurons, and we don't worry about these too much in this course until the end when we talk about the eye. But where we see these are in our special senses. So our special senses are going to be the eye and the ear and the vestibular system, or our balance, as well as our olfactory nerve, or our nerve of smelling. So seeing, smelling, and hearing, those are our special senses. Just know for that, here's the bipolar neuron with that cell body in the middle and the different parts of it. Okay, now we're going to talk about myelination. So myelination is a structure that's going to wrap around individual neurons, and we create this structure called a myelin sheath. And the myelin sheath basically just speeds up that signal so things can happen much faster. And we'll go into more detail with that. But our myelinated neurons for this course are sensory neurons. So every sensory neuron that we focus on in these traces are going to be myelinated, and that will affect kind of the pathway they can take. And then we have preganglionic motor neurons that are going to be myelinated as they go out into the periphery. In reality, interneurons are also going to be myelinated, and there are some skeletal motor neurons that are, but right now, all we need to worry about is these two types of neurons are myelinated. And then we need to know that non-myelinated neurons for our class is going to be the postganglionic motor neurons. So again, the pre- and postganglionic motor neurons are used in autonomic nervous system traces. And I'm saying a lot of words now, but once we get into all this, it will make more sense and it'll be super clear. Just kind of to illustrate, there are two neurons. One is myelinated, one is not. Myelin is created by Schwann cells, and it wraps individual axons like this, and it creates this white appearance. So here's our signal that's going to travel down the axon, and this is just going to show you the speed difference. So you can see the little duck got all the way to the end way before the top one. So that's just basically what myelin does. Speeds things up. All right, cool. So now we're going to look at structures of spinal nerves. So now we're getting more into gross anatomy type stuff. It's harder to see neurons. And when I say gross anatomy, I don't mean disgusting cadaver juices dripping on your shoe. I mean larger structures that you don't need a microscope to really visualize. So we can see this structure. What we're looking at here is the central nervous system, specifically the spinal cord. And within the spinal cord, there's these two types of matter. In the middle, we have that butterfly, or H, looking shape, that's called the gray matter. And the gray matter is where cell bodies will be housed and where synapses can occur. And then everything around that H or the butterfly is called white matter. And white matter is big myelinated tracts. You don't need to worry too much about that for this class, but we will talk about what runs in there. The way I remember this, because I always mixed them up when I first started, is that looks like an H, so I would call it the Grach matter, like gray H, Grach matter. Yeah, this is pretty stupid, but it works. There is something where a sensory neuron is talking to a some type of motor neuron, and that's called a synapse, and we can see those synapses are going to occur in the gray matter. For Mark's anatomy class, you don't want to draw that synapse in the white matter. You always want to keep the synapses within that gray matter. So now we're kind of looking at a diagrammatic image of the whole spinal cord. So the spinal cord is really long. Each one of these little nerves coming off the spinal cord is representing a bunch of levels. So I'll show you what I'm talking about. Here's that top level. That's representing C1 through C8. So 8 levels up in the cervical region. So what that translates to is cervical 1, and then 2, 3, 4, 5, 6, 7, 8. So that top little nerve coming off the spinal cord, that's C1 through C8. That's just the basic layout of C1 through C8. Here's the next level. So after C8 we hit our thorax, and we're going to have 12 thoracic levels. So T1 through 12, we have 12 thoracic levels, and then we also include the L1 and L2 for lumbar 1 and lumbar 2. This picture that corresponds with T1 through L2 is kind of the basic pattern of those levels. So if you compare C1 through C8 to T1 L2, you can notice one tiny variation, and we'll talk about what that is if you don't notice it. We go down to the next one, and we're going to see L3 to S1. This one kind of looks like C1 to C8, but in real life it's just lower. Then we hit this level, S2, S3, S4. And we can notice there is one difference here compared to the L3, S1, and it's that big nerve hanging off, and we'll point that out here eventually. But that's the basic pattern of S2, S3, S4. And then we're going to hit S5 to the coccyx, the first coccygeal nerve. So just there's two nerves there, S5 and coccyx 1. Those are two nerves, and that's the basic pattern. So basically we have this basic pattern throughout the entire body, but T1 through L2 and S2, 3, and 4 are where our modifications occur. And if you can remember, T1 through L2 was for sympathetics, S2, S3, S4 was for parasympathetic, and that's why these are different, and we will go into that. So just pointing out some of these, that yellow arrow just traced out the dorsal rootlets or posterior rootlets. Those are going to correspond or insert into the posterior part of the spinal cord. So if we were to orient this, those rootlets are going to the back or posterior part of that spinal cord. So the nice thing about this is if we have a dorsal rootlet, we're also going to have ventral rootlets. And those are going to come out of the anterior or ventral surface of that spinal cord. Next, we're going to have this here. So continuing from the dorsal rootlets, that whole, I can retrace that. Looks like I have to go through all my slides again. Let's get back there. There's our dorsal rootlets. Here's our ventral rootlets. And now next we're going to have the dorsal root. So rootlets get larger and we create this thing called a dorsal root. So just a larger rootlet basically. If we have a dorsal root, we're also going to have a ventral root. But first, before we get there, we're going to look at this structure. So we have all these blue balls on the dorsal roots. And we can see every single dorsal root has that at every single level. Anytime we see a bulge like this on a nerve, we call it a ganglion. And any ganglion that we're going to find is going to have cell bodies in it. And if we can remember what type of cell bodies we have, we're going to have pseudo-unipolar cell bodies, or we're going to have multipolar cell bodies. So when we're talking about this ganglion, which we call the dorsal root ganglion, because it's a ganglion on the dorsal root, so the dorsal root ganglion, this is where the pseudo-unipolar cell bodies are going to reside, or this is where they're found. So if you can remember those little balloons, that is where we'll find those cell bodies. And then we have the central process of that going into the spinal cord and the peripheral process going out to the extremities or wherever else. But those ganglion on the dorsal root, the DRGs, dorsal root ganglions, those only house sensory neurons. And we can see that ventral root, there is no ganglion on there, which means there's no cell bodies housed in that ventral root. Once the roots come together, they're going to split, and there's this branch here. That's called the ventral ramus. The word ramus just means a branch. So this is the ventral branch, or ventral ramus is what we call it, ventral ramus. And this is going to go to our hypaxial muscles and our limbs, basically anything that's not our epaxial region or our very back part of our body. So that's our ventral ramus. If we have a ventral ramus, we also have this little guy hanging out here. That's called the dorsal ramus, and that's going to go to our back muscles, which we call the epaxial muscles. So ventral and dorsal ramus. A couple other things to label in here. So every level we can see these tiny little nerves hanging out, coming off the ventral ramus. Every single level has one of these, and this is called a gray communicating ramus. So we have a gray communicating ramus at every level, and we can see that. We can see at T1 through L2, we have an extra little nerve hanging out there. That's called the white communicating ramus. So if you were given this picture without the levels written to the right of this spinal cord, you could identify that level or that picture, that spinal nerve as T1 through L2 because there's a gray and a white communicating ramus. Or there's just two communicating rami, and that does not happen at any levels except T1 through L2, which is our sympathetic level. And that white rami is there for the sympathetic nervous system. And if we go down to S2, S3, S4, we have this guy coming off the ventral ramus. So this this nerve coming off is called a parasympathetic splanchnic nerve. So it's called parasympathetic because it uses the parasympathetic nervous system. Splanchnic refers to going into a body cavity. So this is going to organs because that's where organs are in our body cavities. So parasympathetic splanchnic nerve. This is specifically going to our organs of the hindgut. So for this class, the organs of the hindgut are descending colon, so descending colon, the sigmoid colon, and the rectum, as well as the bladder. It goes through the penis, pretty much anything down there in that pelvic region. A mnemonic to help us remember what S2, S3, S4 does. Some people like to say S2, S3, S4 keeps the penis off the floor because these are the levels responsible for erections. So S2, S3, S4, penis off the floor. It also innervates that pelvis area. So that's a parasympathetic splanchnic nerve. So here's the picture that we're going to be focused on for the traces for the majority of the rest of this presentation. This is the picture we use when we do our traces. So traces, when we're doing nervous traces, are not the columnar blood trace lists. We're just going to be drawing those neurons in. That's why we need to know the shapes and kind of where we find them. So we'll have this picture and we just have to draw a bunch of neurons on it with our instructions. So let's look at this. We're going to zoom in here and we can see there's the spinal cord with the gray matter, white matter. We can see that ganglion entering into the back of the spinal cord, and we know there's a dorsal root ganglion. So there's the dorsal root with the dorsal root ganglion coming off of it. Let's see if I can color on here. Does not look like I can color. But going into that posterior part of the spinal cord, there's our dorsal root with the dorsal root ganglion. Coming out of the front, there's our ventral root. And then they come together, but there's this structure sitting on top of that. And we'll talk about what that is. But everything to the left, or everything lateral to that kind of large tube with the other bulges on it, lateral to that, those are our rami. So where the upper limb box is, the ramus that's going to that is our ventral ramus. The one that's going away from the upper limb box, that's our dorsal ramus. So dorsal ramus goes to our back muscles, ventral ramus goes to everything else. That's an extremity or a body wall. So that big structure that kind of separates the roots from the rami in this picture, we call that the sympathetic trunk. So I'll outline it here. There's the very top of the sympathetic trunk, and we trace it all the way down to the very base. That's our sympathetic trunk. This picture is kind of hard to tell, but it is not in the same compartment as the spinal cord. The spinal cord would be wrapped, not wrapped, but inside of the vertebral canal. So we got our vertebral column, and within it is where our spinal cord is. The sympathetic trunk sits on the outside of that vertebral column. So it's not in that same vertebral canal created by the bones. So two different locations. There's also no connection between the ventral roots and dorsal roots to that sympathetic trunk. The only connection is with that white and gray communicating ramus, and we'll make more sense of that. We can see some bulges on that sympathetic trunk, so there's some lighter blue balls. Again, if we have bulges on a nerve, we call those ganglion. These ones are on the sympathetic trunk, so we call these the sympathetic trunk ganglion. And they are going to house cell bodies, but not sensory, because sensory cell bodies are only in the dorsal root ganglion. And these ones, we're going to have neurons, or neuron cell bodies of neurons called post-ganglionic motor neurons. So post-ganglionic motor neurons are going to be in the sympathetic trunk ganglia. Coming off of that sympathetic trunk, these red arrows are pointing to these. We have these nerves going into body cavities. You see ones going into the thoracic cavity, to the stomach, to that sigmoid colon, all cavities, or all organs in a cavity. These are called sympathetic splanchnic nerves. So we had parasympathetic splanchnics at the bottom, S2, S3, S4 levels, but at all of these levels we have sympathetic nerves going into the cavities. And we know these are sympathetic because they come right off of the sympathetic trunk. So those are connected to the sympathetic trunk. And we can see there are bulges or swellings on those nerves. So we're just going to call these sympathetic splanchnic ganglion. So they're on a sympathetic splanchnic nerve, so we just call it a sympathetic splanchnic ganglion. And again, those are going to house the cell bodies of post-ganglionic motor neurons. And then finally, that green circle at the bottom, where the sigmoid colon box is, this is representing a terminal ganglion, which the parasympathetic nervous system uses. So in that green box, we have another post-ganglionic motor neuron cell body that lives in there, but this is only for parasympathetic traces. These ganglion are found in the walls of the organ, not outside of the organ, but they are inside of the wall of the organ around that smooth muscle. And again, parasympathetic controls the smooth muscle of the organs, so that the neurons are just going to go to the organs and then they synapse in the actual wall of the organ. And that green again is called a terminal ganglion. And you can see on the right there's the levels, so C1 through C8, T1 through L2, L3, S1, S2, S4. And Mark usually does not label these levels on the exam. He expects you to know how to do that. The best way to do that is you can see T1 through L2. There's the two communicating rami, and the larger one is the white communicating rami. Every other level has a gray, so that's the smaller one. And then S2, S3, S4, coming off of the ventral rami, we see that parasympathetic splanchnic nerve going to the sigmoid colon. We can see there's two splanchnic nerves. One comes off the sympathetic trunk, so that's our sympathetic splanchnic. One comes off the ventral ramus, so that's our parasympathetic splanchnic. So to give all this some context, here on the left, we have a vertebrae. This looks like a cervical vertebrae because of the forked spinous process, and there's some vessels going through transverse foramen here. But we can see the spinal cord, see the vertebral body, so we can orient anterior, posterior. Coming off, we can see the bulges, which are more lateral. And then moving more laterally, we can see these tiny little communicating rami, which would be leading us to that sympathetic trunk. So that's just kind of a cross-sectional view of a single level. On the right, we have a body cavity, stomach's cut away, we have the kidneys. We can see these yellow nerves with the ganglion. There's our sympathetic trunk with the sympathetic trunk ganglion on them. And we can see those are on a separate area, not next to the spinal cord. So they're just not in the same compartment. Same type of context here. We can see I took away a vertebrae, and we can see the spinal cord with the rootlets coming off. And then leading out, we can see where the sympathetic trunk is with the ganglia, and we can see two communicating rami. So there's the connection between the ventral ramus and the sympathetic trunk. So again, key thing here is the sympathetic trunk is not by the spinal cord. There's no connection there. All right, so we'll get into nervous traces here, and we're going to start with the somatic traces. So again, somatic traces are controlling skeletal muscle, and we use skeletal motor neurons. So anytime we're using a skeletal motor neuron, that equals it's a somatic trace, and that means we're going to skeletal muscle. These are pretty basic. You use a sensory neuron in to the central nervous system, and use a skeletal motor out. To refresh your memory, this is what sensory neurons look like. This is what a skeletal motor neuron looks like. And we use one more type of neuron in these somatic traces. All the traces can use these. They're interneurons. Interneurons just connect all the levels together, as well as opposite sides of the body. So interneuron means between neurons. So this is just going to communicate between one neuron and another. There's this terminology, ipsilateral. Ipsilateral just means the same side as the stimulus. So if I'm getting burned on my right hand, the ipsilateral side is my right side. Contralateral is just the opposite. So if I'm getting burned on my right hand, the contralateral is my left hand. Here's some rules that we need to know. So sensory neurons always enter the spinal cord using the dorsal roots, and their cell bodies are always in the dorsal root ganglion. For this class, always is 100% correct. So sensory neurons will only and always go in through dorsal roots to get to the spinal cord, and the cell bodies are always in the dorsal root ganglion. You do not draw their cell bodies anywhere else. Skeletal motor neurons always exit the spinal cord through the ventral roots, and their cell bodies are always in the gray matter. So we can see the synapse happens between sensory and motor in the gray matter in the spinal cord. And then interneurons have to be used to cross over to the contralateral side or the opposite side, or to travel up and down the spinal nerve levels, or go up and down the spinal cord. But you can't do both at the same time for this course. So if I need to go to the contralateral side, I have to use an interneuron to cross over before I can go out of the spinal cord. if I have something at my foot, that spinal cord level, um, if I have something at my foot spinal cord level and it causes me to do something with my upper limb, that sensory is going to come in way down there, and then an interneuron is going to travel up all the way to my cervical region, and that's where it can send the signal to my upper limb. So interneurons just connect different levels together. And here's how we're going to draw these with the colors corresponding. So one thing to know, sensory neurons can split to communicate with other neurons. So here's one input there on the left communicating with 3 outputs. So that looks like it's communicating with 2 skeletal neurons and an interneuron. So if you have to do one thing, or if you have one stimulus that's causing multiple things, the sensory neurons can do this. Interneurons can also do the same thing. So there we have a sensory neuron synapsing with the pink interneuron, which we see splitting 3 times. Again, it's communicating with all types of neurons. And that's just so it's not one-to-one all the time. It's like one sensory can communicate with a bunch of different things. And that's just more logical. And here's what interneurons kind of rules for that. Interneurons can move up and down levels or cross to the opposite side. You can't do both at the same time. So if I need to go up and cross, this is what it looks like. So you just want to think of them as an elevator, and then you have like the moving walkway bridges. They just go in one direction, up and down, or horizontally, never at the same time. So here's an example of a trace. And this is a somatic trace. You're going to have an input, integration, output. So here's a baby, here's a pin. What we're going to do here is we're going to put a sensory input, and we're going to see the reaction. So it's going to happen pretty quick, just pay attention. There's the sensory, there's the blue going in, sensory neuron. There's the motor going out from the central nervous system to move that muscle and pull that arm away. After that happens, which is a reflex, consciousness happens, and then you know you just got hurt. But that happens after your body has already moved. So here's an example of a trace. Just kind of a trace that Mark will give you. A stimulus means a sensation, to the upper limb initiates a skeletal muscle response in the contralateral upper limb. So just translating this, a sensation in my upper limb is going to cause a muscle response in my opposite upper limb. So I can see that I have 2 upper limb boxes. What I like to do to keep these organized, especially at first, is I put an eye next to upper limb as my input, and then I put an eye next to that upper limb box. And that will just keep things in order for me. So I know I have to start there with a sensory neuron. And then my output, or the end of my trace, is the contralateral upper limb. And I put an O next to that box. So I know I need to end in that box. And that's what my trace needs to look like. So I need to start on one side and end on the opposite. So I kind of need to think to myself, what do I need to do here? And I'll show you. We're going to use these neurons, sensory, skeletal motor, interneuron. Again, sensory should be a pseudo unipolar shape, but just diagrammatically, this is just for the colors. So the stimulus is picked up in the upper limb. So there's my fork, the receptor, and that neuron is going through the ventral ramus. It's the ventral ramus because it's a limb. You would only use the dorsal ramus if it's your epaxial region or your epaxial muscles. So then it's a sensory neuron. I know it has to go through the dorsal pathway now because we're at the roots. The dorsal root is a one-way street for sensory. The ventral root is a one-way street for motor. So that blue is going to go through that back nerve. There's the cell body in the dorsal root ganglion, and you have to draw it like that with the little popsicle stick. And it forks in there so it can communicate with a another neuron. So just to double check myself, I started in the right place, I got it into the central nervous system, and my cell body is in the dorsal root ganglion. Okay, I need to get over to the other side, so I know I have to use an interneuron to cross over. I can't just continue that sensory neuron over. I got to use an interneuron to cross. So I'm going to draw that synapse just like that. Now I took it to the other side, and I can communicate with the skeletal motor neuron, which I know is going to exit the front or the ventral part of that spinal cord through the ventral root. And you can see the blue and the red kind of stops right there. That's because that's the sympathetic trunk, and there's no connection there. This is just representing the sympathetic trunk is kind of anterior to that structure deep, which is called the spinal nerve trunk. So the sympathetic trunk is anterior to the spinal nerve trunk, and that nerve trunk is where the ventral and dorsal roots come together. So as they come together, they create a trunk, and then they split into the dorsal and ventral rami. But we can see that that motor neuron went out the ventral ramus to that upper limb, and that would be the trace there. So I started and ended in the right place. Okay, so here's one you'll try. I've attached a PDF of the nervous trace paper. You'll want to try this one. You can pause the video whenever you want so you can see this trace. All right, so hopefully you did that by yourself. I'm going to walk you through how I would do this. So translating this, sensation from the lower limb initiates a skeletal muscle response in the ipsilateral lower limb, the ipsilateral upper limb, and the contralateral upper limb. So one input is causing three things to happen. One in the same box, one in the box up top, and one in the opposite box on the top. So both upper limbs and the same lower limb. Again, I like to use the eyes. There's my input, and there's an O for that one, O for that one, O for that one. Here's the types of neurons we're using, and this is a somatic trace because we're going to skeletal muscle. We're moving skeletal muscle, so it has to be a somatic trace. So here's the input, in from that lower limb, through the ventral ramus, into the dorsal root. There's the cell body in the dorsal root ganglion, and there's two branches there. That is because I'm doing one thing. I have one output to that same lower limb box, and then I have two outputs way up high, way up in the cervical region. So one of the branches I'm just going to use for a skeletal motor neuron, which goes straight to the box, and that trace is done. So I check it off. Now I need to get up to those corresponding levels, C1 through C8 for those upper limbs. So the way I do that is I take an interneuron all the way up, and that's how you would draw it on your exam. You just keep it on that same side, and you head it up, just drawing through the other spinal cord levels, up to the levels you need to get to. And you can see I have two forks. Again, I have two outputs up there, so I gotta get to both of them. So on the left side, there's the skeletal motor neuron, and we get to the upper limb box there, and that one's done. Now I need to cross over. This is similar to that other one. I just take another interneuron to cross over, bridge over, and then I can go out the front, and I affect that upper limb. So that may look like a lot. It's pretty basic there. You just need to keep in mind interneurons can go up and down or cross, never at the same time. Okay, now we'll get into autonomic traces, and all that's really different here is the specific rules we need to know, but other than that, these are pretty basic. So here's the types of neurons we use in autonomic traces. Again, we're going to have sensory, we're going to have interneurons, but then instead of skeletal motor, we use these neurons called preganglionic and postganglionic motor neurons. These are still motor because they're going to muscle tissue, but they're going to smooth muscle instead of skeletal muscle At least the postganglionics are. Preganglionics actually meet up with postganglionics and synapse with those, and we'll see how that happens. So we have two different types of traces, since that autonomic nervous system is composed of two different subsystems. We have the sympathetic. So sympathetic, again, is an autonomic trace. It's going to smooth muscle of blood vessels. So any blood vessel in the body that has smooth muscle is going to be controlled by the sympathetic nervous system. That means I could say, take me to a blood vessel of the stomach. And the key word there is blood vessel. Even though we're going to where the stomach is, we're still just going to a blood vessel. So if it's ever a blood vessel, just think sympathetic. If I said, take me to the smooth muscle of the stomach, that would be different, because now we're going to the smooth muscle of the stomach, not a blood vessel of the stomach. Hopefully that makes sense. We use preganglionic and postganglionic motor neurons. Again, we're doing smooth muscle, so it requires two motor neurons. The preganglionic motor neuron output has to come out of T1 through L2. So this is critical. You need to be able to identify T1 through L2 spinal levels. And again, those have the two communicating rami. But when you draw the preganglionic motor neuron, its cell body has to be up at that level. It can't come out of anywhere else. We're going to be able to use sympathetic structures, which means a sympathetic trunk, the communicating rami, and those sympathetic splanchnic nerves. The pre and post ganglionics are communicating with each other, and that communication or that synapse is going to happen at the last possible ganglion, or the ganglion closest to the box we're trying to end in. And that will make sense when we get into this. And here's a little fact that we need to know. The neurotransmitter commonly used by sympathetics is norepinephrine. You can also say epinephrine, it's totally fine. But just know norepinephrine or epinephrine is the neurotransmitter released. We like to call the sympathetic our fight or flight nervous system. So it allows our body to get blood to where it needs to go or run away. Fight or flight. A couple more rules. We always use three neurons. We have the sensory in, and we have the preganglionic motor neuron to a postganglionic motor neuron. Always the three neurons. You can also have interneurons, but this is the general pattern you need to know. Sensory, pre to post. You have to know this. I stress this a lot. Preganglionic sympathetics always come out of T1 through L2. So if we look at this picture of on the left, we can see T1 through L2, where those two communicating rami are, the white and the gray. That spinal cord level just a little bit superior to it, that is our T1 through L2 spinal cord levels. So any preganglionic has to come from those levels. Preganglionics can and will go through white communicating rami. So when we draw those, they can go through the white. They do not go through gray. And that's because they're myelinated. So if you can remember, sensory neurons are also myelinated. So sensory neurons can also go through the white communicating rami. Postganglionic cell bodies are only found in two places. Sympathetic trunk ganglion, so that long tube, the sympathetic trunk, those ganglion on there house postganglionics, or the sympathetic splanchnic ganglion on those sympathetic splanchnic nerves. That's also where we find postganglionic cell bodies. And interneurons can be used when needed. So if I need to affect a blood vessel in my neck and in my lower limb and in my stomach, you'll see interneurons being used. So here is a sample. I will walk you guys through this one. So stimulus from the thoracic cavity, we're just going to say my lungs or my trachea, etc., initiates a sympathetic response to a blood vessel in the thoracic cavity. So something in my thoracic cavity is causing a blood vessel to do something. So that's translating that. It doesn't matter if it's constricting or dilating. Sympathetics can do both. but we're just going to a blood vessel. So I'm going to zoom in, and we're going to put our eyes. So thoracic cavity, there's my input in that box. My output is a blood vessel in the thoracic cavity. So I need to start and end there. There's the types of neurons we're using. Remember, we always have 3: sensory, pre, and post. So that sensation comes from the thoracic cavity box, and we go through that sympathetic splanchnic. You can see I did not draw a cell body in that ganglion. That's because sensory cell bodies are only in the dorsal root ganglion, which we see right there. There's the cell body for the sensory. So it went through the sympathetic splanchnic, onto the sympathetic trunk, through the white communicating ramus, because it's a myelinated neuron, and that connects us to the ventral ramus. And then we leave a little gap there because that ventral ramus, that area, that spinal nerve trunk is not connected. And then we go through the dorsal root because that's the sensory input pathway. Now we use this 2-neuron pattern. We have a preganglionic and a post. Pre means before, post means after. So we're going to use the orange neurons synapsing in that gray matter going out of the front. Again, preganglionics are myelinated, so that's going to go through the white communicating ramus, and we get all the way to that sympathetic splanchnic ganglion. That was the ganglion, preganglionic. Now postganglionic is going to be everything after this ganglion, so we're just going to switch to that postganglionic and then end in the box with the fork to get to that blood vessel. So that's a sympathetic trace. Here's another example. So now a stimulus from the epaxial. You have to know what epaxial means. It just means your back. So you're going to learn about epaxial muscles, which are back muscles, or your epaxial region, which is basically a strip of skin in the back, strip of dermis. So a stimulus from the back of the abdomen initiates a sympathetic response to a vessel in the epaxial abdominal wall and to a vessel in the sigmoid colon. So for some reason, something on my back is causing a blood vessel of my organ to do something, as well as a blood vessel in the same spot. So we'll just go through this. Input, there's our epaxial abdominal wall. Output, we know we're going back to that because we're going to a vessel there, and we're also going to have an output in the sigmoid colon, that box there. The key thing to remember here is it's a sympathetic trace, and there's 2 nerves or 4 nerves going into that sigmoid colon box. So we need to know which ones. And since it's sympathetic, we should use the sympathetic splanchnic, which is connected to the sympathetic trunk. So we need to keep that in mind. So we're going to just do this trace here. Here's our stimulus from the epaxial wall. You can see I went through the dorsal ramus because I said epaxial. Dorsal ramus innervates epaxial. And then we go through the regular pathway, dorsal root with the cell body. And now I need to do 2 things here. So I'm going to take an interneuron up. And what is the reason I did that? So think to yourself, why did I go up there? Why didn't I just have 2 forks of the sensory? Well, why I did that is I have 2 outputs, and they're both going to a blood vessel. And the only way I can go to a blood vessel is if the output originates at that T1 through L2 level. And that level is T1 through L2, where that interneuron traveled up. And I know that because of the 2 communicating rami right there. So that interneuron traveled up and then split up there. So now I'm at the correct level for the preganglionics to come out of and eventually get to their end targets, which is a postganglionic, which gets to the final blood vessel. So here's one part of the trace. There's our preganglionic coming out of the front, and it's going to go through there, through the white ramus, and there it ends. It splits right there in that sympathetic trunk ganglion. We use the sympathetic trunk ganglion to go to our body wall, so epaxial abdominal wall. It could be our cervical wall, it could be our thoracic wall, anything like that. It's just not a body cavity. So anytime we're not going to a cavity, we're going to use the sympathetic trunk ganglia. Same thing with the limbs. So we can see that preganglionic is going to synapse with a post, and postganglionic neurons are the only ones that can go through the gray, communicating rami. And you can see I went through the dorsal ramus. I kind of backtracked there, but you have to do that because we're going back to an epaxial blood vessel. If we were going to a hypaxial or a blood vessel anywhere else, then you would use the ventral ramus. But we're going back, so we have to go through the dorsal ramus. There's no connection there between ventral ramus and the epaxial. Cool, so check mark that output. Now I need to get down to that sigmoid colon. So what I'm going to do here is cross over. And this just kind of keeps things cleaner. But I'm going to go to that side. And now I'm just going to bring that preganglionic as close to that box as I can. I'm going to find the ganglion closest to that sigmoid colon box, and that's where my synapse between pre and post is going to happen. So there's our preganglionic going out, again, through that white ramus. We're bypassing the sympathetic trunk ganglion, and we get to a sympathetic splanchnic ganglion. Again, preganglionic synapse with postganglionic. There's that synapse, and now we are at the blood vessel of the sigmoid colon there. We're not going to the organ, we're going to a blood vessel that's just found in or around that organ. Alright, so now it is your turn. Feel free to watch this weird video, read this trace, and see if you can do this on your own. Alright. Yeah. Okay, so here's that trace. In the description here, I will make a key for this trace. Alright, now let's jump to parasympathetic traces. So parasympathetic traces, very basic. There's only one possible one he can have you do. That's purely parasympathetic. He can mix parasympathetic and sympathetic together, and we'll kind of walk through that. But here's the parasympathetic traces. Parasympathetic is another type of autonomic. It's going to smooth muscle of the gut tube. It uses a sensory neuron in, uses pre and postganglionic motor neurons out. But the difference here is the preganglionics come from the vagus nerve or S2, S3, S4 spinal levels. Sympathetics was T1 through L2. Parasympathetic is S2 through S4. You do not ever use sympathetic structures with the parasympathetic nervous system. Sympathetic nerves are for sympathetics, not parasympathetics. And then the pre and postganglionic synapse happens in the wall of the organ, or just the box that says hindgut or sigmoid colon, just the very bottom box. Its neurotransmitter is acetylcholine. You'll want to know that one for the parasympathetics. So neurotransmitter is acetylcholine. And we call this our rest and digest system because it's really active during rest, and it causes peristalsis of the GI tract, so digestion. So the vagus nerve innervates our foregut and our midgut organs. The foregut is the esophagus, the stomach, and the duodenum or duodenum. The midgut is the jejunum, the ileum, the cecum, the ascending colon, and the transverse colon. So all of those organs are innervated by the vagus nerve, which comes all the way from the head. And then we have our hindgut, which is going to be innervated by the S2, S3, S4 pelvic parasympathetic splanchnic nerves. And our hindgut is the descending colon, sigmoid colon, and the rectum. So here's a possible trace. This is a vagus nerve trace. I haven't seen one of these before, but just keep in mind it is possible. There's the sensory neuron, there's a dorsal root ganglion way up there, and you synapse up in the brainstem. I wouldn't worry about this image too much. Just be aware vagus nerve controls the foregut and the midgut. Here's our pelvic parasympathetic trace. So this is the bottom level, S2 through S4, and we know that because of the ventral ramus has a splanchnic nerve coming off of it. We can see the sympathetic splanchnic nerve coming off the sympathetic trunk, and we can see the parasympathetic splanchnic nerve coming off of the ventral ramus. That only the parasympathetic splanchnics are only at S2, 3, and 4. So that's how we know that's the level we're at. This trace is going to be an input from the sigmoid colon, controls the taenia coli, or affects the taenia coli or the smooth muscle of the sigmoid colon. So there's our sensory neuron from the sigmoid colon. We know it always goes into the back. Then we're going to have the preganglionic, which goes all the way to the organ, to the box, and that's where the synapse happens because the ganglion are in the actual wall of the organ. So that's the parasympathetic trace, and that's really the only one that's purely parasympathetic that I have seen Mark test on. All right, so here's a couple traces. It was taken from old exams. Feel free to pause the video. The 3 colors represent 3 different traces. You should be able to do all of these, and I will post the key for these in the description as well. The ones that say that red one is really common on exams, so definitely know how to do that one. Here's some more exams, or some more old questions. And you always want to make sure you follow the directions, especially when it comes to color coding and where you start and stop. Always ask a TA if you're confused. Don't just start coloring stuff in.